Hi, welcome to another lecture on prokaryotes, Understanding Biology Chapter 23. In this lecture, you'll be learning about prokaryotic cell structure and prokaryotic classification. At the end of this lecture, you should be able to describe three basic shapes of prokaryotes, explain the difference between gram-positive and gram-negative bacterial cells, characterize the internal structure of prokaryotic cells, and explain the methods used to classify prokaryotes. Let's look into prokaryotic cell structure. Prokaryotic cells are relatively simple and can be categorized based on their shell shape. Prokaryotic cells exhibit three basic shapes. Most prokaryotes exhibit one of these three basic shapes, such as rod shape, often also called bacillus. In plural, we call it bacilli. We also have coccus shape, in plural, which called cocci, which is a spherical or ovoid shape, as you can see here. And finally, we also have this shape called spirillum. In plural, we call it spirilla, which is a long and helical shape. Spirogyra usually have this spherical shape. So these are the three basic shapes of prokaryotes, and the most important contributor for their shell shape is actually their cell wall. If they do not have a cell wall, these prokaryotes will not have a set shape. So now that we started talking about cell wall, bacteria can have variety of other different structures such as flagella and pili. Let's first talk about cell wall. So bacterial cell wall is often complex that contain many layers. Bacterial cell walls contain a unique polymer called peptidoglycan, which is made of a polysaccharide cross-linked to a peptide side chains. These polymers form a rigid network that maintain the cell shape and present cell that prevent these cells from bursting in hypotonic solution. So they actually have two functions for the bacteria. One is they maintain their cell shape, and they actually help them to withstand these hypotonic environments. And when you look at archaea, they have a similar molecule which we talked in last lecture, that they have pseudomurine, which is very similar to peptidoglycan. So based on the cell wall structure, we can actually divide bacteria into two categories. So we can actually divide bacteria into gram-positive and gram-negative. This can be identified based on this gram stain. Basically, gram-positive bacteria contain a thick peptidoglycan layer, while gram-negative bacteria contain a thin peptidoglycan layer, but also a polysaccharide layer on the top of that. During the gram stain, gram-positive bacteria will appear purple, while gram-negative bacteria will appear pink. So let's first look into the staining process and the principle of gram stain. So when you look at the gram stain, first thing you need to do is you have to prepare a thin smear of bacteria and stain first with primary dye called crystal violet. So both gram-positive and gram-negative bacteria will absorb this dye and become purple in color. The next step is we add grams iodine, which is a mordant, and it will actually make a crystal violet and iodine complex that formed inside the cell, and it will keep both the gram-positive and the gram-negatives in purple in color. This third step is actually the called where we washing these cells with alcohol. This step is very important. The alcohol will wash away the lipopolysaccharide layer of gram-negative cells, and they will lose this purple complex that made with iodine, while alcohol will make thick peptidoglycan to be jelly-like structure, which prevent this purple color from escaping from gram-positive cells. Therefore, they will retain the purple color at the end of this decolorization step, the step 3. In the next step, we are actually counterstaining these cells with another dye, which is safranin, which is a red color dye. And when gram-negative bacteria, which is colorless due to losing this purple dye, crystal violet, they can now absorb this red safranin. And gram-positive bacteria already have their color, so they will remain purple in color, while gram-negative bacteria will remain, now will appear pink in color. So this is the theory behind gram stain, and this method of gram stain is actually used in many identification processes as the first step to differentiate these bacteria into either gram-positive or gram-negative bacteria. So further, if you look into the cell wall structure of gram-positive and gram-negative bacteria, the gram-positive bacteria, the peptidoglycan forms a complex network around the outer surface of the cell, which also contain these lipoteichoic and teichoic acids, which protrude from the cell wall, as you can see here. So here is the plasma membrane of gram-positive cells, and you have this thick peptidoglycan layer which forms a network around the cell wall, and then you have these lipoteichoic acids that are protruding from these peptidoglycan layer. And while gram-negative bacteria, which is this picture here, they have this, here is the plasma membrane, and they have this very thin peptidoglycan layer that is sandwiched between the plasma membrane and another layer which I talked to you earlier, which is a lipopolysaccharide layer. And this layer is actually very unique to gram-negative bacteria, and it enables them to be resistant to many antibiotics. And gram-positive bacteria are susceptible to antibiotics, while gram-negative bacteria, because of their lipopolysaccharide layer on the top, they are resistant to antibiotics. Now that we're done talking about the bacterial cell walls, let's move on to other different structures found in bacteria. So some bacteria can have additional layer on the top of the cell wall, which is gelatinous, and this layer is called capsule. This capsule surrounds the cell wall and enables prokaryotic cells to adhere to the surfaces. This feature is often considered as a virulent factor, as it gives an added advantage to the hosting bacteria, allowing them to evade host immune system by interfering with recognition by phagocytotic cells. So usually when you have capsules, they have a potential to become disease-causing bacteria. So in this picture you can see here is your cell wall in purple color, and on top of the cell wall you can see the capsule. And another two structures that you can find exterior of bacteria is these flagella and pili. Many prokaryotes have either flagella or pili or both. These are cell exterior structures that involve in locomotion and aid in attachment and horizontal gene transfer. Flagella are actually slender, rigid structure, and they actually composed of this protein called flagellin. Flagella can range from 3 to 12 micrometer in length, and they are very thin, only 10 to 20 nanometers in thickness. And they can spin like a propeller and allow cell to move. On the other hand, pili are numerous in number, as you can see here in this picture. They are short hair-like structure. They are found only in some gram-negative bacteria, and their major importance is in adhesion and then movement. They are also involved in this process of horizontal gene transfer called conjugation, which we will talk in our next lecture. Some prokaryotes are able to produce endospores. which are survival structures that allow them to be able to enter to dormant stages until the condition become favorable. Endospore-forming prokaryotes can develop a thick wall surrounding the genome and a small portion of their cytoplasm when exposed to stressful environmental conditions. When environment become improved, like better environmental conditions arrives, these spores can germinate and become vegetative cells. The spores are very tough. They can survive long time without anything. So bacteria such as disease-causing bacteria such as tetanus, botulism, and anthrax actually are able to produce these endosperms. And even though you actually cook or boil these endospores, they are very resilient to heat. So therefore, they can still survive boiling and cooking to certain stages, and they will still be able to come out of their dormant stage and cause diseases. So now that we discuss about the bacterial cell exterior structures, let's look into bacterial cell interior structures. Prokaryotic cell interior lacks extensive functional compartmentalization similar to when we talk about eukaryotic cells. We have a lot of internal complexity because of our endomembrane system. We have membrane-bound organelles. But compared to that, prokaryotes are very simple. They do not have this interior endomembrane system or extensive compartmentalization. So when you look into the interior of bacterial or prokaryotic cells, there are several different interior structures. The first one we are looking at is internal membranes. So many prokaryotes possess these invaginated regions of the plasma membrane that is used in respiration and photosynthesis. So you can see in this picture invagination of the plasma membrane that is used in photosynthesis here and respiration. And we also have nucleoid regions and ribosomes as cell interior structures. So the nucleoid region, like we talked in our last lecture, they are actually prokaryotes do not have a nuclei. Because of that, their single circular double-stranded chromosome is actually condensed in the cell, actually creating this nucleoid region with other proteins and RNA. So this region, this visible region, is called nucleoid region. So they may also contain plasmids as well. The next interior structure that prokaryotes contain is ribosomes. They are smaller than eukaryotes, and they also differ in protein and RNA content. Prokaryotic ribosomes therefore can be targeted by antibiotics. But these same antibiotics will not affect the eukaryotic ribosomes. Therefore, when you treat for infections, we can easily treat bacterial infections with antibiotics. These antibiotics will specially target ribosomes, and they will prevent protein synthesis in bacterial cells, which will eventually kill them. Now that we done talking about bacterial structural features, let's look into a little bit more about classification of bacteria and prokaryotes. In the past, we actually use morphological characters to classify prokaryotes. But in part 1 of this lecture, we did talk about how more 90 to 99% of prokaryotes that are on Earth has not still be identified or characterized. Therefore, just now we are learning into and understanding their biochemical and metabolic characteristics. So these early systems that used these different stains, gram stains, and phenotypic characteristics may not represent the true evolution relationships among these prokaryotes. But nowadays, we have new developments of genetic and molecular approaches that we use in prokaryotic classification. It may help to reflect the true evolutionary relationships of these prokaryotes. These molecular approaches include photosynthetic or non-photosynthetic, finding out if these organisms are photosynthetic or non-photosynthetic, motile or non-motile, unicellular, colony forming or filamentous, formation of spores or division by transverse binary fission, importance as human pathogens or not. So these are some of the observable phenotypic characters. But let's look at some observable molecular approaches that we use actually. So molecular approaches actually include looking at amino acid sequences of key proteins. This is actually a very good approach because protein coding genes are actually very conserved. And if you look at amino acid sequences of key proteins, you may be able to find out relationships among these bacteria or prokaryotes that we are classifying. And another thing that we actually look at as a molecular classification is present GC content. The analysis of nucleic acid base sequences by establishing the presence of guanine and cytosine is another approach. And we also use nucleic acid hybridization, which is essentially the mixing of single-stranded DNA from two species and determining the amount of base pairing. So the amount of base pairing will increase based on how closely these species are related to each other. And we also can use gene and RNA sequencing, especially if you remember when we classify 3 domains of life. Carl Woese actually used a system of comparing ribosomal RNA sequences because these ribosomal RNA sequences are conserved and present among all the organisms except for viruses. Therefore, we can actually use for bacterial on prokaryotic classification, we can use ribosomal RNA sequences. We also use whole genome sequencing, which is also more elaborated. And we find out that larger scale sequencing of random samples, that we find out that this majority of bacteria and a lot of prokaryotes have never been cultured or studied in detail. So the classification system actually allows us to understand evolutionary relationships of these organisms, and most widely accepted classification system is presented by Berg's Manual of Systematic Bacteriology, and this is published in 5 volumes, and you can find this online easily. And this is actually looking into how to classify bacteria into different categories based on molecular data. So in summary, today you learned that prokaryotic cells are relatively simple and can be categorized into 3 groups based on their cell shape. There are a variety of cell structures in prokaryotic cells, including cell wall, capsule, flagella, and pili. Based on the cell wall structure and gram staining, bacteria can be divided into gram-positive and gram-negative. Endospores are cell survival structures, and prokaryotic cell interior is very simple compared to eukaryotic cells, and they contain internal membranes, nucleoid regions, and ribosomes. Prokaryotic classification is based on molecular approaches, and it might reflect their true evolutionary relationships. With that, this is the end of the lecture. I hope you enjoyed this lecture, and let's see again in another lecture. Thank you.